is the 27th. Aki, not here. Boggs, not here. Bonner, if I remember correctly, is online. Bonner, Green, Aki. Yeah, Aki is also online. Again, if you're online, don't forget you have to send the words, otherwise you're not considered present. I know I keep saying it, but we have a repeat offender. Anyway, Aki is online. Boggs is not here. Bonner is online. Again, if you're online, send the words. Clay is Clay? Clay is not here? Okay. Dawson is here. Ellis is here. Paula is here. Green is online. Again, don't forget to send your words before 9 a.m. Harrison is here. Henson is here in red. James is here. Are you in red? Okay, it's hard to tell. Thank you. Kesterson is here in red. King is not here. Lewis, not here. Linda Q, not here. Morrison is here. Grace, if I remember correctly, is online. Don't forget to send your words before 9 a.m. Ward is not here. Wilcox is not here. Wilder is here. And Parks is here, right? No. Oh, yeah, Parks is here. Yep. Parks is here. Did I miss anybody? All right, I'm trying to pull up the PowerPoint. Let's see if it works. So it's been screwing with settings, so I have a feeling I might be fighting for my hands here. Fighting with technology. The first word for attendance, on that note, it's fight. Yep, because I'm fighting with technology, so here we go. I'm not even going to spend time fighting it. I'm just going to work with it like that. This is so annoying.
You said twice the first word?
Yep. Are you kidding me? I don't think this is going to fix it, but I'll try. Oh my goodness, it actually worked. It's a Friday afternoon. All right, so let's get it mostly worked. It didn't completely work because it didn't take us back to where we left off. So let's go. Does anybody remember where we left off? I think I do. I think that was it. Yep, this is exactly where we left off. All right, so to remind you, again, this chapter is about photosynthesis. This is the last chapter before the next exam. We will stick with the exam schedule. So, if we're ahead of the game, for example, we're not supposed to take the exam until like next week or whenever it may be. I need to take a look. I don't remember. Then we'll stick with that, right? So the due date won't change. If the due date was 2 weeks from now, then the due date's going to stay 2 weeks from now. However, once we finish this, I will open it up. So it might be opened up early. That's the only thing that would change. The due date will not change. So again, once we finish this chapter, you will then have all the information you need to take the next exam. So you can take it right away if you want, or you can wait until closer to the due date. Actually, I recommend not doing it right away, right? Take those quizzes as many times as you can, because those quizzes are here to help you for those are your study guides. And remember, once you score a high score in your quiz, no matter what you score after that, your score is not going to change, right? So if you get the full 20 points and then you take it again and get everything wrong, your score is still going to be 20 out of 20. So take advantage of that. I think there were some other announcements I want to tell you before we jump into this. Oh yeah, Veterans Day. I plan on sort of giving myself a day off for Veterans Day since the university won't. So you can go ahead and plan on this. November 11th, I will not be here. We will have class, but it'll be online. So we'll meet online, but we're not going to physically meet here. Of course, I am here for you guys, so if one of you happens to be opposed to that and really, really wants to have class, then I will forego my Veterans Day celebration. You just send me an email and I'll make that happen. But right now, that's my plan.
We go online instead of going to syllabus or somewhere else?
Yeah, it's on syllabus, but yeah, it's a great question. And it is on syllabus, and I will send an email out like probably that Friday before. But hey, don't forget, we're meeting online on Monday, and here's the link to use. Great question. And of course, since we're just meeting online, you could also just watch the video. But whatever. What are some other announcements? So yeah, lab, don't forget, if you weren't here for lab on Wednesday, you check your emails. You need to take care of the video with the questions go along with it and the virtual lab. Oh yeah, all of you, check your emails. I have reason to believe only one of you has read my most recent announcement. And if you read my most recent announcement, you'll know why I think only one of you has read it. Check your emails. That's a big thing. Of course, you guys are doing great anyway, so I guess in this case it doesn't matter much. But usually I have classes with people failing, and I'm like, why don't you check your emails when you're failing? But you guys are all doing pretty good, but still, check your emails, check the announcements. There's important information out there. Any questions about labs or anything before we jump back into photosynthesis? All right, let's jump back into it then. So again, we're talking about photosynthesis. Most of this portion of the exam is going to be a lot like the portion of the exam for respiration, where I say, in which stage is this produced? In which stage is this used? In which stage does this happen? In which stage does that happen? These are the things you're going to need to know. But to understand how it truly works, that's the stuff I'm about to give you right here. There might be a few questions on this section for this next bullet point, but mostly what I'm doing is just trying to give you enough background information so you can understand how things work. And this next bullet point, the nature of sunlight, will help you answer the pre-lab questions about the sunlight, right? The color of light, excuse me. So let's jump into it. The nature of sunlight. Be back out really quickly and make sure we're still recording. I've been having bad luck with that recently. Still recording. Okay, good. Anyway, there we go. The nature of sunlight. Sunlight is a type of energy called radiation. More specifically, it's called electromagnetic energy. So when you use X-rays, when you use microwaves, when airplanes are using radar, when you're using infrared to look at stuff, when you're listening to the radio, this is all, or using your cell phone, right? These are all the same, it's all the same type of energy. It just happens to be different energy. Well, we'll jump into it. It's all the same type of energy. It is different. We'll talk about how. This type of energy acts as waves. I'm not going to ask you that question, but there you go. If this was a physics class, the fact that it acts as waves would be important. We will talk a little bit more about waves here in the next couple of slides, but generally speaking, it's not important for understanding what I'm going to teach you after this portion. So, in my opinion, it's not that important. Speaking of which, the distance between the crest of two waves is called a wavelength. You can see it right here, right at the bottom. There's a crest, that's the wavelength. In this particular example, this wavelength is 580 nanometers, and that's what yellowish orange light looks like. Of course, that's just an example. Obviously, if you're studying, you do not need to memorize that particular color is 580 nanometers. I'm not going to ask you the definition of a wavelength, but you should understand what it is. That way, if I use the word, you understand what I'm saying. You definitely don't need to memorize anything about the spectrum. Like, I'm not going to ask you a question and say, which one has more energy, infrared or UV? Or, which one comes after infrared, microwaves or radio waves, right? I'm not going to ask those questions. So when you're studying, there's no need to study this graphic. It's just a visualization of what we're talking about. Speaking of this graphic and this visualization of what we're talking about, the full range of radiation is called the electromagnetic spectrum. And again, I'm not going to ask you that as a test question, but it is important to know what it is. That way, when I use that word, you know what I'm talking about. So, are there any questions about this slide? All right, the next word for attendance, I'm going to underline it. So, I'm not going to say it. So, if you're online and watching this, maybe you're listening to it, open your eyes and look. The next word for attendance is underlined right now. Are there any questions about this slide before we dive a little bit deeper into these concepts? All right, next concept. When light shines on a pigment, certain wavelengths, i.e. colors, are absorbed and disappear. Meanwhile, others are reflected. So for example, this shirt that I'm wearing right now, it's absorbing all the colors of light except one. Which color is my shirt not absorbing?
Red.
Red. Perfect, thank you, right? It's absorbing all the colors and it's reflecting red back to me. His shirt's red too. Her shirt's white. So her shirt, or part of it, her shirt is reflecting all the colors back, right? His shirt's black mostly. So his shirt is absorbing all the colors and essentially reflecting nothing back. So that being said, what color, let me ask you this, first of all, what color are most plants, like leaves?
Green.
All right, so what color are they reflecting? If their colors are green, then
They're
what color?
reflecting green.
They're reflecting green. Therefore, what color do plants not absorb?
Green.
Green. So keep that in mind when you're formulating your hypotheses for the for the lab, right? Green is the one color that plants do not absorb. That's the one color that they're reflecting. That's why they look green. Also, keep in mind, and I guess I should have made this more clear. White is all the colors. So when you see a white light like this right here, this is all the colors mixed together, right? So when you think about the light that you're going to give the plants, and white is one of them, that's all the colors mixed together, right? And then some other things to consider, because sometimes students do this. Technically, you can see here, it's not a great visualization, but this side of the spectrum is higher energy, and this side is lower energy. So sometimes people make hypotheses based on that. They think, okay, well, blue light's going to be the best because it's higher energy. Or they might say green light is the worst because it reflects, that's the one color that's not absorbed.
How do you know that?
How do we know what?
Which side is higher? You just know that, or you can know from that thing?
Me personally, I know it just because I was taught it. That's what you mean. I don't know how to.
I'm just confused because the numbers are lower on the side that you said was higher.
Good point. Okay, so how do I explain it? All right, so imagine if you and I were holding a rope, right? And I'm doing this nice and slow, not much energy. There'd be big old waves, right? Big waves with like really long wavelengths. Then imagine we're doing that. There's gonna be a bunch of short ones. So the shorter the wavelengths, which is this, right? That number represents the distance between the wave crest. The shorter the wavelength, the higher the energy. And that's why. And it really is just like that. Like the more energy you put into it, quicker it's moving, therefore the closer those are getting together. Same thing musically. You know, like 440 hertz is the wavelength of the air vibrations. I think that's an A. And then 880 is an A also, but it's the next octave up. So things to consider. Great question.
Between blue and red, blue is the better choice to grow plants in. Is that the reason why? It's because the wavelength is more compact?
For what?
The wavelength.
I mean, the blue's better for what? I'm sorry.
Blue is better for growing plants.
We'll see. That's the question we'll find out. That's what we'll do with this experiment, is find out. So yeah, that is one thing to consider. Another thing to consider that we don't, and you don't need to know this for the exam, but just things to consider for the exam. It's really weird. Sometimes these higher energy things don't penetrate things as well as other things. And your textbook doesn't even talk about that. For example, no, that's not a good example, is it? You can look this up for independent work because I don't remember the exact details. But when you go underwater, like when you're scuba diving, colors start disappearing. And I forget which end disappears first. I think it's this. I think because these have lower wavelengths or lower energy. I think first you start like the color red starts disappearing, and then the color orange, so on and so forth. It's worth looking up for independent work. That's one of the few times you can write about something that's not exactly biology, right? Because we're talking about physics, but you can look that up. Which is also interesting. Like when you think about a fish that lives 100 feet underwater, and you describe what it looks like. What's it really look like? You know, if you caught the fish and you're looking at it, it looks different when you're in the atmosphere than when you're 100 feet under the water. But anyway, I digress. That was a fun digression, though. So again, keep all this in mind when you're making your hypotheses. Green is the one color that's reflected. Blue is a higher energy color. White includes all the colors. Just things like that, things to consider. And speaking of which, I'm not really going to talk about this. This is from your old textbook, but this bullet point here is basically what you're going to, it's a different version of the kind of experiment that you're going to do in class, right? In lab, that color experiment. Someone did it already in the 19th century. They did it a different way, but they did do it. And you can look at it if you want. It's downloaded here it's on this PowerPoint. This is again from your old textbook, and usually I delete things from your old textbook unless I find that they're worth saving. So I saved this one. You can read it. I'm not going to test you about it. I'm not even going to talk about it. I'll just show you very quickly. Again, it's very similar to what you did or what you're going to do next Wednesday, except in this case, instead of using bacteria, or excuse me, using pH to measure the rate of photosynthesis and respiration, they literally this guy just put a prism and put a rainbow on this thing where he's growing stuff, and he used this stuff as photosynthetic bacteria. He just found a bunch of them were growing here, a bunch of them were growing here, and very few were growing here. Right? So same way of he's testing the same hypothesis that you guys are doing in that last experiment of the exam, of the lab, excuse me. He's just doing it a different way. So again, for the lab report and the lab, there's questions that say, how else can I test this hypothesis? Think about things like this. Get creative. Also think about all that stuff I said in the B-Lab lecture, where I said if glucose is going up, respiration must be happening. If glucose is going down, photosynthesis must be happening, right? So if you could somehow find a way to measure the levels of glucose, you can measure the rate of respiration of photosynthesis. So get creative. The experiments don't even have to be real, as long as you can just come up with something hypothetical, like I just said. If we can measure the rates of glucose, but then of course you need to come up with some sort of experiment to do that. Anyway, we went off track, or I went off track a little bit too long. Let's move forward, unless anybody has questions about this slide right here. Okay, if you download the PowerPoint, you can watch this little animation. It shows the relationship between light and pigments. So again, the pigment that we're dealing with is chlorophyll. It's green. The reason it's green is because it's absorbing all the colors other than green. Excuse me. So let's talk about these chloroplast pigments. The selective absorption of light by leaves is why they appear green. So everything we just talked about, everything I just said, right there it is in writing for you. The selective absorption of light by leaves is why they appear green. In other words, once again, green light is poorly absorbed, therefore it's reflected towards the observer, and that's why it appears to be green. Something else to consider, as you know from the last chapter, first law of thermodynamics. Energy cannot be created or destroyed. So that means when that light energy hits these pigments and it's being absorbed, it's not disappearing. Energy can't just disappear, right? It has to be converted to another form. So let's talk about that very quickly. Actually, before we even talk about the next slide. When it's really hot outside and bright, and you don't want to get hot, what color shirt should you avoid wearing? Black, right? Good. Because black is absorbing all the colors. And what's happening, and I'm not going to get into this deep in the exam, but this will help you understand how photosynthesis works a little bit. It's absorbing that light energy. And what happens is, and I'm going to say this again when you can see a slide of it, it's exciting the electrons, right? You know, your shirt's made up of atoms, right? Atoms have electrons. That sun energy excites those electrons. But then those electrons can't stay excited, so they just come back down to the regular state. And when they come back down to the regular state, they put off heat energy, right? So that that light energy was then converted to heat energy, right? So we think of the sun as hot. I mean, the sun itself is hot. But when that light is hitting us, that's not technically the heat. The heat is coming from us absorbing it and our electrons getting excited and then losing that excitement and putting off the heat. I would take questions, but just let that sit in your head for a bit, and we'll come back to that. So again, a visualization of everything we said. Here's the sun, some source of light, any source of light. It's white light, right? So all the colors are hitting it. All the colors are being absorbed except green. Green is reflected back. That's why it appears green to our eyes. Here's where it gets a little bit trickier. I'm going to put a question mark next to this because I'm not sure if I'm going to include that on the exam, but there are 3 different types of pigments. So earlier in this chapter, I told you there's chlorophyll, and that was a true statement. However, now I'm getting more specific. Technically, there's 2 different types of chlorophyll, and there's also something called carotenoids. We're about to talk about them right now. Chlorophyll A, that participates directly in the light reactions. So remember, photosynthesis is broken down into light reactions in the Calvin cycle. It's chlorophyll A that directly participates. They all these things participate in light reactions. Obviously, they're pigments, right? So they're going to be the things that are acting with light. The chlorophyll A is the one that does it directly. If anything, that's the bullet point you need to know. Yes, technically it absorbs mainly blue, violet, and red light, but I'm definitely not gonna ask you that. Long story short, and I'm gonna go ahead and give you a spoiler here. For each one of these, I'm gonna tell you what they absorb. It doesn't matter specifically to you guys, this one absorbs this one absorbs that, this one absorbs this, that doesn't matter to you guys. Point being, all together as a team, they absorb a lot of different light because they all absorb different different light. So anyway, that's chlorophyll A. It's the big one. It's the one that mainly or directly is involved. Then we have chlorophyll B. It absorbs mainly blue and orange light. And again, I'm not going to ask you that. Blue and orange. I like the Gators. I don't like the Gators. Morrison, do you like? What are your thoughts on the Gators? Do you even care? Because you're a Hurricane fan, right?
Yeah, but
Yeah, not much. Okay. I think didn't the Hurricanes beat? Yeah, you guys beat Florida the first week. Yeah, good stuff. Anyway, so chlorophyll B, the more important part. I don't really care what it absorbs. The more important that it conveys its absorbed energy to chlorophyll A. So again, remember, chlorophyll A is the main pigment there. Chlorophyll B is just absorbing energy, and it doesn't directly participate. It's just like, oh, I've got this energy. Let me hand this off to chlorophyll A because chlorophyll A knows what to do with it. Any questions about chlorophyll B? All right, here's the one that's, as far as the exam is concerned and as far as these experiments are concerned, this is probably one of the least important ones, but as far as our everyday life is concerned, this one's actually a little bit more important. Carotenoids. Carotenoids absorb mainly blue-green lights. Again, not that you need to know that. Their function is to absorb and dissipate excessive light energy that might damage chlorophyll. So in a sense, the carotenoids are the sunblock for the leaves. So once again, think about this, and I've been hinting at it, now I'm finally going to say it. When we do these experiments, that first experiment to see how does light intensity affect photosynthesis. Common sense will tell you the more light there is, the more photosynthesis there is, right? Then if you think a little bit deeper, just like everything else I've taught you in the semester, there is too much of a good thing. So light is a good thing, but obviously too much light isn't a good thing. It's not good for us. We get sunburnt. It's not good for plants either. This is why they have things protecting them. So there is a point where there is just too much sunlight. Of course, that depends on the individual plant and the individual species and the environment. But yeah, there is a point where there is too much sunlight. More importantly, or more, again, this is very unimportant, but more relevant to what's going on today, carotenoids, let's talk a little bit more about them. That's where we get our fall colors. Actually, I'll come back to that. First, let's talk about them being human nutrients. I'll put a big old X through this because I'm not going to ask you anything about this. But it's interesting, I think. Beta carotene is one of the carotenoids. It's a orange/red pigment. So when you see pumpkins or sweet potatoes or carrots, that's where they get their orange color from. And that is converted to vitamin A in the body. Which reminds me, a little side note here that has nothing to do with what we're talking, or little to do with what we're talking about. There's a type of GMO rice. I think it's called super rice. And people are all afraid of GMOs because I don't think they understand how GMOs work. So what they did, in some places in the world, people eat rice, and that's almost all they eat, right? They're poor, they don't have access to good nutrition. So what some scientists did was figure out how to take the gene that makes beta carotene, they took it and they put it into the rice genome. So now we have this rice that now produces beta carotene, right? It's good for them, it's good for them. It's helping people survive. People, again, are afraid of GMOs because I don't think they understand how they work. But that's all they did. They took a gene that was found in carrots and they put it in rice, and now rice produces beta carotene. Or, and again, I'm getting off on a tangent here. Another way people don't understand GMOs, there's a type of apple that doesn't turn brown when it's exposed. When you cut an apple and leave it out in the air, it turns brown. Well, this one doesn't because it's a big, scary GMO. All they did was go in there and there's a gene. There's an enzyme responsible for making it turn brown. So they just took that gene out, right? It's just a regular apple. It's just missing one gene. But anyway, that's beta carotene. Lycopene is a bright red pigment that you find in tomatoes, watermelons, red peppers, right? Anytime you see red, not anytime, but a lot of times when you see red in a fruit or a vegetable, that's where it's coming from. It's lycopene. It's an antioxidant. It's really good for you. And again, a big X through that line, or through that slide, indicating I'm not going to ask you any questions about that on the exam. Interesting stuff to look up for independent work if you want, though. Like beta carotene, right? I've listed some places where you can get beta carotene. Where else can you get it? I said it's converted to vitamin A in your body. Well, what does vitamin A do? Lycopene. I gave you some examples of where you can find it. Where else can you find it? I said it's an antioxidant, but what is an antioxidant, right? I also said lycopene is a potential anti-cancer properties. How? What is it? How is it doing that, right? Those things you can look up for independent work. And again, what I was trying to get at earlier, why this is relevant today, because technically we're in fall now, but this is where the fall colors come from. The fall colors are all those carotenoids. What happens is in the fall, all the chlorophylls leave, the green stuff leaves, and this is what's left. So right now, when you're seeing a green tree, those carotenoids are there, we just can't see them because the green is blocking it. So once that chlorophyll leaves, then we see all the fall colors. Any questions about this that I'm going to put index through, because again, I'm not going to ask any questions about fall colors. All right, so earlier I was telling you, actually I'll just back it up and show you again. Remember we were saying that chlorophyll A absorbs this, chlorophyll B absorbs that, carotenoids absorb this. So here's a visualization of what we're talking about. I'll also put a big X through this, indicating again that I'm not going to ask you about it. But again, the point is, like I was saying earlier, even though you don't need to know the specific wavelength that each specific pigment absorbs, I'm just showing you that they work as a team, right? So this one does really good at absorbing this wavelength. This one does really good at absorbing this wavelength. This one's the only one pretty much that's absorbing that wavelength, so on and so forth. So as a team, they absorb more. Which makes me wonder, and this, I guess you can't really write it, maybe you could write an independent work topic about this. Maybe this has been looked into, I don't know. What if one day evolution results in yet another pigment? What if there's some other pigment and now all of a sudden we can start absorbing green, right? Obviously plants wouldn't be green anymore, but wouldn't that be interesting? Then plants can be more productive. Then we can pull more carbon dioxide out of the air. Then therefore, hopefully we might take care of our climate change issues. But anyway, any questions about this slide? All right. Now this next bullet point is more important for the exam. I'm gonna give you some information that you're definitely gonna need for the exam. That last bullet point was information that'll help you understand some things I'm about to teach you. The things I'm about to teach you mostly are gonna be on the exam. Not this first slide though. This first slide, this is just for you to know. Your book talks about it, so do I. Like I said earlier, light, that electromagnetic energy, it behaves like waves, like I said earlier, and I said it wasn't important, and it's still not, but I'm bringing it up again. But it also behaves as discrete packets of energy called photons. I'm not going to ask you that, but I am definitely going to use this word photons. So you need to know what a photon is. It's a fixed quantity of light energy. So when I say an electron absorbs the energy from a photon, that's what I'm talking about. Right? Photons are that light energy. And again, like we mentioned earlier, thanks to Bree asking about it, the shorter the wavelength, the greater the energy of the photon. Not that I'm going to ask you that, but it is true. This picture was from the old textbook. Yeah, obviously that person's just putting on sunblock. I don't even remember why your old textbook used that. Probably had something to do with like, how many of you heard of UV light, ultraviolet? It's really, really short. It's shorter than the light that we can see. UV light we can't see. But it's really, really short. It's really, really powerful. And that's where we wear sunblock. We're protecting our skin from the light that we can't even see, right? But anyway, here we go. I've already said this earlier when I was talking about wearing a black shirt out in the heat. But now I'm going to put it in writing. Let's talk about it again. When a pigment molecule absorbs a photon, the pigment electron gains energy, right? So again, the black pigment in your shirt, when you go out and it's being hit with light, that light in it has energy. The photon is absorbing that, or excuse me, the electrons are absorbing that energy. They're gaining that energy. The electron is now excited, and you've heard me use that word a lot, right? High energy electrons, AKA excited electrons. I keep saying that, right? Because there is a difference between high energy electrons and just regular electrons, right? They have a lot of energy. So that's what happens. Light hits the pigment molecule, the electrons get excited. However, the excited state is very unstable, so it can't stay that way. So instead, it usually loses that excess energy immediately and falls back to the ground state. Again, remember, energy can't be created or destroyed. So when I say it loses the energy, it's not like the energy just disappears. It has to be converted to some other form. And again, usually what happens, at least, again, using the shirt example, that's usually converted to heat energy. However, photosynthesis takes advantage of that energy. So instead of things just absorbing light energy and then releasing it as heat energy, what's happening instead is they're taking advantage of that high energy, right? They're making these high energy electrons, and instead of letting them lose that energy, they're going to use it. So let's talk about how that happens. First of all, here's a picture of what I'm talking about. And this is, again, this is a picture from your old textbook. It's not my favorite because, yes, part of this is definitely relevant. Light hits the chlorophyll molecule. That's exciting the electrons. The electrons cannot stay at that excited state. It's very unstable. So they just lose that excitement and come back down to the ground state. Again, energy cannot be created or destroyed. It has to be converted. In this case, in this picture anyway, this is why I don't like this picture. In this case, it's converted to heat and light, which again, that's not really what we're after, right? Because that does us no good for photosynthesis. We want to capture that energy. So yes, when it's not being used for photosynthesis, that light or that energy can be released as heat or light. But that's not what really happens. Also, this is sort of worth mentioning, this next slide right here. Some emit light and heat after absorbing photons, which is, again, that's what that picture is, right? It absorbed the light energy from the photons. And in this picture, for some reason, it's instead of taking advantage of that energy, it's releasing it as light and heat. And this is, again, this is from your old textbook. I only kept it because I think it's slightly interesting. It's not really important at all for the exam. But I'm sure you guys have seen glow sticks before, right? The light emitted by glow sticks is caused by a chemical reaction that excites electrons. So what happens is, I don't know if you ever used a glow stick, it's not glowing at first, right? How many of you have ever used a glow stick and made it glow? Like it wasn't glowing, then you made it glow. How'd you make it glow?
Shake it.
Yeah, shake it, break it, right? Because what's happening is there's chemicals in there that aren't mixing. And then once you crack it, then the chemicals mix, and the chemical reaction happens that's exciting those electrons. But in this case, once they lose that excitement, again, that energy can't disappear. So in that particular case, it's released as light energy. And that's how that works. And I don't know this for a fact, and you could look it up for independent work. And again, this is one of the few times where you can look up something that's not technically biology. But I've always heard that if you store those things in the freezer, like after you've already started them, after you've broken them, I've heard that if you store them in the freezer, they glow longer. I don't know if that's true. I imagine if it is true, the reason would be because obviously, usually the colder things are, the slower chemical reactions are happening. Therefore, the slower chemical reactions that are happening, the longer you'll have fuel. I'm just speculating, I don't know. That would be interesting to look up for independent work. Anyway, back on to more important stuff. In the thylakoid membrane, remember I told you I would use that word, so you need to know what it is, right? In the thylakoid membrane, the chlorophyll molecules are organized with other molecules into something called photosystems. And much like thylakoid membranes, I'm not going to ask you what photosystems are, but I will use that term, and that term will be used in the exam, so you need to know what they are. Again, I'm not going to ask you the definition, but I'm going to use that word. So each photosystem has a cluster of a few hundred pigment molecules. Again, I'm not going to ask you that either. This is just so you understand what's happening. Let's show you a visual of what we're talking about. Here's an entire chloroplast. We're kind of zooming into this one little thylakoid membrane right here. You can see it's a double member phospholipid bilayer like we talked about a long time ago. Not really relevant to this specific conversation. But here we go. That's a photosystem, right? That's one photosystem. So you can imagine there's a bunch of photosystems embedded there. This is just one photosystem with hundreds of pigments, molecules in there. So this cluster is like a light gathering antenna. It's the words from your textbook. I would say, I think a better word to use would be to say this cluster is like a solar panel, right? Because it is the thing that's absorbing that light energy. So let's talk about how that happens. First of all, a photon strikes the pigment, and I kind of skipping. There's a little bit I'm skipping between these two bullet points, so let's talk about it. The photon strikes the pigment. That excites the electrons in the pigment. Again, that's a very unstable state, so they can't stay that way. But in this case, instead of it coming down from the excited state, it actually jumps around from molecule to molecule. until finally it arrives at the reaction center. I'm not going to ask you to know that the energy bounces around from molecule to molecule until it finally arrives at the reaction center. All you need to know is that this light is hitting these pigments and it's exciting the electrons, right? So these details I'm not worried about, but it's in your textbook, so I wanted to include it. That way when you're reading your textbook, you could remember, oh yeah, I remember him saying that. I remember him saying he's not going to ask about that. So anyway, it makes it to the reaction center. And the reaction center, as you might guess, based on what I said previously a few minutes ago, it consists of chlorophyll A. Remember, that's the primary pigment in this. The chlorophyll B and the carotenoids, they're just there to help chlorophyll A. So then, the chlorophyll A molecules that sit next to the primary electron acceptor traps the excited electron. So again, that's the whole thing, right? Again, photons hit the electrons, or the pigments. That excites the electrons. That excitement bounces around. That's not so important. The important part here is that excited electron is basically captured by the primary electron acceptor. And then another team of molecules uses that trapped energy to make ATP and NADPH. So here's something that you already know when we did the overview, right? And that's a test question. Which of the two stages produces ATP? Light reactions. Which of the two stages produces high energy electrons, which are carried by NADPH? Light reactions. So that bullet point right there, that's what we're after. That's the most important bullet point as far as we're concerned. We're exciting these electrons. That's what the sun does. It excites the electrons. And then forget all that middle stuff. Who cares? That excited electron is eventually captured, turned into, and then that energy is then used to make ATP and NADPH. Everything else in the middle, as far as I'm concerned, doesn't even matter. You need to know that light hits the electrons, it excites the electrons. That excited electron is eventually captured, and the energy from that electron is used to make ATP and NADPH. And in a sense, and you're going to see soon, it's very similar to respiration. Because remember, in respiration, we had a high energy electron, and then we let it release its energy, and as it released its energy, that energy was used to make ATP and NADPH. Well, in that case, NADH, but high energy electrons, right? So we'll come back to this. The next word for attendance is going to be slide. Reminds me of the electric slide because we're talking about electrons. Not that I like that song. I think I'm one of the few people who hate that song. I also don't like choreographed dances. Anyway, we're getting off on a tangent again. So again, here's everything we just said, but in one big picture. Once again, we're zooming into this chloroplast. We're zooming into this thylakoid membrane right here. We're zooming in and looking at one of many photosystems that has hundreds of molecules. More importantly, again, light hits it, it excites the electron, and that electron is going to be captured and used to make ATP and NADPH. That's the big thing, right? That's what you need to know. We are exciting, that's what the sun is used for, is to excite the electrons. And again, that's something else you need to know for respiration and photosynthesis, right? So photosynthesis, what are the ingredients? Well, we need water, we need carbon dioxide, and we need sunlight energy. And you need to know how those three things are used, right? So right now I'm telling you how light energy is used. It's used to excite electrons. Because we need excited electrons to make ATP. And then to jump ahead to what I'm about to tell you later, we need ATP to squish those carbons together to make glucose. Right? So this is all in process to make glucose later. Anyway, any questions about this before we dive deeper into this? All right, then let's dive deeper into this. How the light reactions generate ATP and NADPH. Technically, there are two photosystems. I'm not sure, and if you guys have done the pre-lab, you already know all this because you've watched Hank Green describe this in a more entertaining and quicker way than I did, but let's talk about it anyway. I'm putting a question mark because I'm not sure I'm going to differentiate in the exam between the two photosystems. I do want you to know what they do as a team. Altogether, you need to know what they do, but I'm not sure I'm going to get specific. Like this one does this one does that. But let's talk about it. First, again, like we already said, photons excite the electrons in the chlorophyll of this first photosystem that we're going to talk about, right? So again, electrons get excited. Like we already said, the electrons are then captured by the primary electron acceptor. And again, I'm not going to ask you, like there will be no test question where I say, when the photons excite the electrons, what captures the excited electron, right? I'm not going to ask you that. However, I might say, in which of the two stages does the primary electron acceptor capture high energy electrons? In which case you should say it's the light reactions. But again, you do need to know that the electrons are excited and they're trapped. I'm just telling you right now, I'm not going to be so specific where I ask you the name of the thing that captures them. Anyway, so here we go. An electron just left, right? We excited an electron and it was taken away. Now electrons have to come from somewhere, they have to go from somewhere. If you take an electron away, one has to be replaced. So the photosystem replaces that lost electron because water donates it. And that's also something I've told you before, and there it is again. Water is the thing that's donating the electrons. And when water does donate those electrons, that's where we get the oxygen from. I told you this before, I'm telling you it again, that will definitely be a test question. For this exam, whether we're talking about photosynthesis or respiration, those three things should always be together in your mind. Oxygen, water, and electrons, right? Because here, water is donating these electrons, which produces oxygen. And then in respiration, it's the opposite. Oxygen accepts those electrons, which becomes water. And again, test questions on this slide. Which of the two stages is associated with photosystems? Light reactions. Of course, that should be really easy because photosystems and light reactions, right, that gives it away. Which of the two stages excites electrons? Light reactions. Which of the two stages excites electrons? Did I just say that? Yeah, sorry. Which of the two stages uses chlorophyll? Light reactions. Which of the two stages uses water? Light reactions. Which of the two stages produces oxygen? Light reactions. What is the purpose of water? To donate electrons. A lot of test questions in this one slide. And I'm hoping this should be easy for you. A lot of it should be. Because when you think of light reactions, you should think light and energy, right? And this is all about energy. This is all about like changing that light energy into a form of energy that the cells can use. Anyway, are there any questions about this slide before I show you a visualization of this slide? All right, here we go. Again, light hits the chlorophyll, it excites the electron, which is captured by that primary electron acceptor. Where those electrons come from, they had to come from somewhere. They came from water. Water donates them, and that creates oxygen. Let me add this. I also want to point this out. You can see here that technically, when water donates electrons, it's 2 electrons. And technically, that results in 2 hydrogen protons and half of an O2 molecule. But guess what? Not going to get that specific. You don't need to know about the hydrogen protons. You don't need to know that it's half of an oxygen molecule. You don't need to know that it's 2 electrons. What you need to know is that water is the thing that donates the electrons, and when it does that, that produces oxygen. Another thing you don't need to know, speaking of which, you can see here, when I say the electrons are excited and captured, yeah, technically that's 2 electrons, but I don't care. Just know that electrons are excited and captured. But yes, they do happen to be in pairs of 2. Any questions about that slide? All right, so let's talk about the second photosystem, or the next photosystem. So again, just remind you what we're talking about here. The last thing we talked about, we have this high energy electron up there. It's been captured. So now what we're talking about is like, well, what happened to that high energy electron that was captured? That's what we're about to talk about. That energized electron that was captured now goes through an electron transport chain, just like the one in respiration. Well, not just like, but as far as we're concerned, just like the one from respiration. It's going to go from protein to protein to protein. And as it's doing that, it's going to lose its excitement. It's going to release its energy. And guess what happens? Just like in respiration, that energy that it's releasing is going to be used to pump hydrogen ions against the concentration gradient, so then they can just diffuse back down the concentration gradient. And when they diffuse back down that concentration gradient, that's where we make our ATP. Just like in, say, the respiration, right? I mean, there are slight differences, but as far as we're concerned, they're basically the same thing. They do the same thing. Excited electrons go from protein to protein, releasing their energy, pumping pumping hydrogen ions against a concentration gradient, so they can come back down to make ATP. But also, remember, in respiration, oxygen was the thing that ultimately accepted that electron, and that converted it into water. But here, that's not happening. After we make this ATP, after it releases its energy to make ATP, we're not done with it yet, because we still need high energy electrons to help build. We're going to incorporate that into our molecule of glucose, so we still need them, right? But what did I just say right here? To make the ATP, it lost its energy, right? The photons excited it, and then it released all that energy to make the ATP, so it's not excited anymore. Therefore, we have to excite it again, and that's what we're going to talk about here. So this is the part we already saw. We already talked about this, right? This is the first reaction. The electrons get excited, they get captured, they go down the electron transport chain, which pumps hydrogen ions against a concentration gradient so they can come back down, which produces ATP. But again, now we have a low energy electron because it's released its energy. And notice too, these aren't on the same, this is not right here, right? This is up a little bit. So it's low energy, but it's not as low as it started. But anyway, it's lower energy than it was here. So it gets hit by light again. It gets excited again. And look, and this is on purpose. It's even more excited than it was here. And then it gets captured again. And this time though, when it gets captured, it then is shuffled off into the Calvin cycle where we use those high energy electrons and we incorporate it into the glucose molecules. As you remember from the conversations about respiration, I kept saying, we break down glucose to make pyruvate. And then we break down pyruvate, right? And we break it down because there's high energy electrons in those molecules. And that's what breaking down those molecules in respiration, we're releasing that energy, right? So we have to have high energy electrons. This is where they came from.
I have a question.
Yes.
So these two will always happen in conjunction.
Yeah.
It's gonna be one or the other.
Correct. Yeah. Another thing I don't remember, to be honest, because now I'm curious now that you're asking about it. I wonder, and I don't remember, I'm sure I was taught this and I feel bad. I've been teaching this so long that I haven't gone back to my roots. I'm wondering, is it like always like these two photosystems always working together? Like this one's always providing that one? Or, I wonder if like in real life, if it's like, all right, this one's providing this photosystem, that electron, and then maybe some other time, some other photosystem is providing the electrons. And maybe this one, I wonder if it ever provides electrons to some other photosystem. I don't know. I don't remember. Sadly, this is being recorded. Hopefully none of my bosses watch this and be like, I taught him that. He should know. But no, I don't remember. Good question, though. Not important for the exam, which is the good news because I'm not going to get that deep in the exam, but great question. Again, the purpose here, what we're doing here is we're creating high energy electrons that we can use to put into the glucose that we build later. And ATP, so we have energy to build that glucose later. Here's a great picture. This is from the old textbook. I love it because it really does a good job, illustrating what we're talking about here. It's the same thing I just showed you, except now it's more like a metaphor, right? We have this low energy electron, doesn't have much energy, potential energy in this case. So we smack it with a photon energy, or kinetic energy in this case, which gives it enough energy to come up here. So now it has a lot of potential energy. They just put it down, gravity does its work, so potential energy is lost and converted into kinetic energy, which produces ATP. Then we need to energize it again. The photon hits it, which in this case is the kinetic energy. It comes up here, we have a lot of potential energy again, and then it just goes into that bucket, and then off it goes into the Calvin cycle, where it's incorporated into a molecule of glucose. And the last word for attendance will be purple. That's it. So we'll see you guys on Monday. I will be on office hours at least until noon. Please let me help you if you need help with this pre-lab. Or if you weren't here on Wednesday and you've got to do the virtual lab and the pre-lab and the video, please let me help you. These are not exams. You're not on your own. Yes, sir.
What was the last word? Sir?
Purple.
Purple.
See how that was messed up? And if you're online, don't forget to send the words. You are absent. You're considered absent unless you send the words. There's one of you who's done this consistently. Of course, my hypothesis is you're sleeping right now, and that's why you've been doing this consistently. So probably preaching to the choir.